Hello, this is Mahanta Didia. This is a continuation of Module 7 related video 2. The Module 7 builds on the information from composition and formulation, and we looked at equipment. Equipment and processes are interrelated. If any of them fails, then product will fail. In this second related video on manufacturing process, we will cover lab batch process written in the literature and commercial scale batch. In this submodule, we will start with batch formula and write that for a commercial use. The FDA guidance and similar guidances from international regulatory are available for the manufacturing. Batch formula, it lists all the ingredients that are used in the manufacture of a representative batch of a drug product. The amounts are in kilos. Additionally, percentage could be in form of weight to weight and weight to volume. It is important for a developmental formulations to use weight to weight, weight to volume, and milligram per dose. This will help technology transfer to commercial batch sizes. Commercial products are at large scale and they are made using weights rather than volume, as it provides better accuracy. Reader is requested to review submodule video 1.3 that covers unit of measure, weight, volume, and density. Batch manufacturing process with unit of measures. This is derived from a patent application literature provided here. The fexofenadine hydrochloride solution formula is written here. This is very well written from a unit of measure perspective. It provides milligram per 5 ml or gram per 5 ml. It also provides weight to weight and weight to volume in each of the column. This helps us to design the commercial batch record much easily. For example, 170 kilos is per 100 liter. And this provides a calculated density of the product. And many of these components have densities that may be different than a literature value, depending on its sources. Formulation includes fexofenadine that's dissolved into polyethylene glycol and propylene glycol. Glycerin is a solvent. Flavoring agent is raspberry flavor, and xylitol and sucralose are sweeteners. Disodium EDTA is used as a chelating agent, and small amount of purified water is used to dissolve water-soluble ingredient. Water and glycerin are miscible. You will notice that there is no preservative used in this formulation. It does not require preservative as water component is very small. You will also notice there is no buffering agent in this. Again, this is mostly made out of organic solvents such as glycerin, polyethylene glycol, and propylene glycol. So that buffering agent is not necessary. The laboratory process flow diagram is based on literature. First step involves dissolve/stir polyethylene glycol and propylene glycol stir in 30 minutes. So this kind of process can easily be accommodated in a glass beaker on a hot plate with a magnetic stirrer. Add fexofenadine hydrochloride and stir until clear solution is obtained and continue stirring for 24 hours. This is reasonable for laboratory process. Glycerin is added, which is the largest component, and at this point the beaker will have a lot of solution present, and glycerin is denser, so that the magnetic stirrer used will require higher RPM to mix the solution. A separate tank is used to dissolve aqueous water-based solutes. Sodium EDTA, sucralose, and xylitols are dissolved in water. The water mix is transferred into the main tank and is stirred for 1 hour. The resulting solution contains acid. This is then transferred into PET bottle. On a lab scale, this process is reasonable. Let's look at pilot scale processes and working volume. As mentioned on equipment, working volume is extremely important when we use a larger scale batches. And this will become evident in this slide. Making 1 kilo beaker and magnetic stirrer is acceptable. Pilot scale at 100 kilos is a different story. Main tank for making pilot batch of 100 kilos will require 85 liter working capacity. That means the tank total volume about 150 liter would be acceptable, as it would fit between 40% and 70%. In this case, it is 56%. We take 150 liter tank. The polyethylene glycol, propylene glycol, and fexofenadine mixture is approximately about 4.1 liter. This may not be acceptable in a conventional mixer, as a conventional mixer's propeller may not reach the liquid level to mix the drug. Only 4 liters of liquid is present in a 150 liter total volume mix. That's too small. That's why we should calculate volumes actual or estimate based on density and weight. This information is required at each step. Based on that, we should select the equipment with acceptable working volume. We will look at that at a 1000 kilo batch. Process and working volume at each step. And this should be done at developmental stages. So you build your developmental batches using commercial type processes. For each component, specify weight, volume, and density. And many times, density is estimated, and that's acceptable for small volume. Density is equal to mass divided by volume. At each step, look at the maximum working volume and the containers that can be used based on 40 to 70% of the total volume. And it may depend on the product. It include a rinse step for each active that is transferred, product potency. Let's look at the batch formula for 1000 kilo. First step is to make active. Dissolve the active in PEG 400 and polyethylene glycol. Glycerin, we use that as a rinse step if we were to transfer this. Item 5 and 6, which are glycerin and flavors, are added, followed by separately mixing aqueous phase, which has xylitol, sucralose, sodium EDTA, and purified water. So now we have divided steps into active drug solution, rinse step, add glycerin and flavor into the main tank, prepare separately aqueous phase solution. So instead of having everything prepared in a main tank, we make active solution in a tank that provides optimal mixing conditions. We mix in the main tank with glycerin from item 1 to 6. Aqueous phase tank, we provide the aqueous and transfer that into main tank. Formula as given, percent weight to weight. Then calculate weight per 1000 kilo. So for example, weight to weight percent is 0.51. We will need 5.1 kilos per 1000 kilo of an active. Similarly, for glycerin, percent weight to weight is in 2 parts, 2. And let's focus on 83.5. We will need 832 kilos per 1000 kilo batch. And subtotal at each step. Step 1, subtotal is 34.9 kilo. At step, when we add items, 1 through 6, the weight is 889.5. So this is your main tank. Active gets added to the main tank. Total weight of active solution plus glycerin and flavor is 889.5 kilos. Aqueous phase is made on its own. Total weight is 110.5 kilos. So each step, convert that into volume by using volume in liters equal to mass over density. And the volumes are listed in parentheses. Total volume of the batch is 799.68 liters. In this case, the density of glycerin as 1.26 based on the literature value. Actual densities of each of the products or each of the material will be available from the supplier based on their manufacturing conditions. So once we have the volume of each step, that is 32.25, 663.57, 711.69, 87.99, and the total volume of 799, we can now size the equipment and select mixer. Batch formula for rounded off to 800 liter, we can calculate mixer and total volume. Weight at active dissolution is 34.9 kilos. Estimated volume is 32.5. So that the total capacity can range from 46 to 80 liters based on 70% or 40%. And preferred range will depend on the manufacturing side, and 50 liter would be a reasonable. Aqueous phase, 110 kilos. Volume is estimated to be 87.99. Total capacity of mixer is 125 to 219 liters. Preferred would be 200 liter total capacity of a mixer. Solution in main tank. There are several steps that use main tank, which is common. Initially, we start with glycerin and the flavor. That's 834.6 kilos. The volume is 663.57, again based on the density. And the volume range would be 648 to 1659 liters. We need to continue to calculate all the things that will go in that main tank. Decide on a mixer. Next step, when the main tank is used, active is added. It is 889.5 kilos. The volume is 711.69. 40% and 70% range will be shown here, 1017 to 1779 liters. The final product that now contains aqueous phase as well as step 1 through 6. The total weight of the final product in the main tank is 1000 kilos, rounded off to 800 liters. The capacity will be 1142 to 1999 liters. To select tank size so that all these steps can be done in the same tank. 1500 liter as a manufacturing tank. This may change depending on the equipment available. And we may have to adjust. Instead of 1000 kilo, we may have to make a different batch sizes. So we will need 3 tanks, one to dissolve active, another to dissolve aqueous phase, and a main tank. Storage tank is a separate tank, and a 1000 liter tank should be sufficient. So now let's look at the process flow diagram with mixed tank with the weight. And now we will add quality in process sampling at each of the major steps. from a science as well as FDA perspective or regulatory perspective, we want to add in the process flow diagram as well as in a manufacturing description. Step 1 of a drug solution in a 50-liter mixer tank. That's also listed in the batch formula. Start these steps 24 hours before initiating step 2 because there is a mix for 24 hours. So we add and mix PEG and polyethylene glycol. Now these are waxy material. So it's good to heat that at about 40 degrees if acceptable based on pre-formulation study. We'll slowly disperse hexafenadine into the mix. Mixing, no vortex, and mixing will continue for 24 hours. Cool this to 25 to 30 degrees. At this point, in-process checks will be clarity, assay, and weight control. Step 2 is in the final product solution mixer tank. That to be 1500 liters. 832 kilo of glycerin, item 5. Mix for 60 minutes. Mix flavor, item number 6. And mix for 60 minutes. And we continue mixing until the bulk is transferred. So this mixing will continue all the way until it is transferred into a storage tank. So after the completion of step 2, transfer the drug solution. It would be pumped into the main tank. And in this case, we'll use glycerin to rinse it. So glycerin in a small container, add it to the 50-liter mixer and the rinse solution transferred. At this point, we can check the weight after the rinse solution is transferred. The weight should be 634.6 kilo plus or minus 0.5 kilos. The aqueous phase is separately made in a 200-liter tank. First dissolve xylitol and sucralose. They are very soluble. Add EDTA, mix for 30 minutes and check the weight. Transfer aqueous phase solution into the final mix. The weight should be 1000 kilo plus or minus 1.1 kilo. Mix for 60 minutes. Sample bulk for testing according to quality control specification prior to filtration. Filtration is done to remove the particulate matter, not undissolved solutes. Filter the solution through 20 micron filter into bulk holding tank. Test the filter integrity. Sample the bulk after filtration for testing according to the QC procedure and package the product in approved bottles. The equipment train is shown here. The tanks are shown are representative, they are not to the scale. Aqueous solution into 200-liter mixer tank. Drug solution tank, which is 50 liter, that will be transferred. Glycerin rinse container used to rinse out drug solution and transfer lines and put into a mixed container. Aqueous solution is made separate, be transferred. Once the solution is finally mixed, it's passed through a filter and into a storage tank. This type of equipment train, this should be done as early as in development stage during technology transfer and commercialization.